Good morning, everybody. Welcome back. So, week 2 of our school, which is going to be an extremely exciting day. We have really, today, two of the giants of neuroscience. They might not want to hear that, but I think it's true. And so it's Edvard Moser and Giacomo Michelati. Tony will introduce the first speaker, Edvard Moser, today. And Edvard will also speak later this afternoon or at 6 o'clock at the Palace Macaya, which is fairly nearby, close to the Sagrada Familia, in also a public lecture about the science of memory and how this might relate to improving the human condition. If you want to, it's more a lecture towards also a general audience, to which you're also all invited. But first, we have them all to ourselves this morning. And make good use of that. As you know, we discussed it also last week. One brain, one question. Be active. Every slide is wrong. So pose your questions, right? Find out what these things really mean. Make good use of your time and our time. And contribute to the dialogue. That's why we take so much time for our lectures. So let's get going. Tony will now introduce Edvard. And let's have some fun.
Thanks, Paul. So you guys are very, very lucky. I can't believe how lucky you are. Just to be at the school is great, but to be here today is amazing. You know, it's such a privilege for us as researchers, but I think for students, it's this is going to be one of the highlights, I think, of our BCBT series today. And it's a real pleasure for me to introduce our first speaker, Edvard Moser. He probably doesn't need much introduction to this audience. Together with his colleagues and May-Britt Moser and John O'Keefe, they have really made a massive advance in neuroscience and have opened up a whole new way of understanding and discovering the brain. When we look at the brain, we see millions of neurons, and to characterize what those neurons do is one of the core challenges for neuroscience. And we've been trying to do this for decades, even centuries, and we are still struggling to characterize even a small number of neurons and their function. And that gets especially hard as you move away from the sensor and motor periphery and you go towards the center of the brain. And that's why it was so amazing when John O'Keefe in the 1980s or in the 1970s was able to show that there are cells in the hippocampus in the rat that respond to location in space. And that was an amazing thing, to find these cells almost in the middle of the brain, which appear to code for whereabouts you are when you're moving around. But there were still many questions to answer about this system. In particular, we didn't know then whether cells respond to place, but whether they also know anything about distance or direction, and how the code for that representation of space could possibly work. And the contribution that Edvard and May-Britt made was to help us understand that not only do these cells code space, but they code space in a metric way, that there's information in the hippocampus about how the rat is moving, the distance and direction, and from head directional cells as well, we know that there's a metric map or something that gives the equivalent power of a metric map, so that animals, including ourselves, can build up an understanding of space where they know not just where they are, but how far they need to go and in what direction in order to get to some goal. So it's fantastic to have Edvard here. It's actually his second visit to BCBT, which is wonderful. And Edvard is, I think, going to tell us the story of the discovery of grid cells, but also update us on some of the most latest research from his lab in Trondheim, which, if you get the chance to visit, I've been there twice, is a fantastic lab with lots of labs, lots of rooms in which rats are running around exploring in large chambers. And if you really want to understand how to do neuroscience, go there for a masterclass, because there's lots of rooms which have the same setup, but they're running a slightly different experiment. So we heard last week from Patrick Haggard about how to design well-controlled experiments. Well, if you want to do that in neurophysiology, go and learn from Edvard Moser. Edvard.
Thanks a lot, Tony, for nice introduction. Thanks also to Paul for inviting me. It's nice to be back again. I always like to go to Barcelona, but this is also an exciting school. Now, I hope I don't repeat myself too much, but I've tried to focus on unpublished work, so that will be most of my talk. But I still have put in about 10 minutes, 10-15 minutes of introduction just to bring everyone on board. So, as you know, what I consider one of the great scientific breakthroughs during the last few centuries is that we are actually beginning to bridge physiology and psychology and we're beginning to understand complex behavioral phenomena in terms of the neural activity that goes on in the brain. So this is a pretty challenging task, and that explains why it has taken so long to get there. And much of the background is, of course, just the mere complexity of the brain, which includes the fact that there are so many neurons and so many connections between neurons. So an estimate of about 80 and 100 billion neurons in the human brain, and then if all of them have about 10,000 synapses with other cells on average, then you get up to many, many trillions of synapses. And if you then know that information is distributed among them, then the task gets pretty complex. But nonetheless, I mean, there are a few areas in neuroscience where that connection to psychology has been made. You'll hear about two of them today, and my focus will be on one of them, where the focus is on space. So, as you know, and as Tony told, over the last almost 50 years, actually, there has been pretty much progress in understanding the neural basis of space, or at least the mapping part of space, how we map the outside world, our position in space, in the brain. And I will try to bring you into this. This is also a complex task, so finding one's way is not always easy. Sometimes it is because the local environment is not distinctive. Other times it is because there are very few peripheral cues, like some places in the world. But we nonetheless manage pretty well. Even if we blindfold ourselves, we can, at least for some meters and for some tens of seconds, keep pretty well track of where we are. So how does the brain solve this? There are a few brain structures that have received more attention than others. That doesn't preclude other structures from being part of the spatial representation, the spatial mapping and navigation functions of the brain. But there has been much focus on the hippocampus, and more recently also on the strongly interconnected entorhinal cortex. So I will focus on those, but first, how do we approach these two brain structures? So the human brain is not by itself an easy structure to study for obvious reasons. So much of the work has actually been done in simpler systems, and that makes quite a lot of sense because the way that the brain and also the cortex is wired up is not too different when we come to the principles as we go down on the evolutionary tree. So this shows a human brain, a macaque brain, and to the left you have a rat and mouse brain. You can see how the number of neurons decreases pretty significantly from approximately 86 billion in the human brain to only 71 million in a mouse brain. And of course, if we are not particularly concerned about the cortex, but just about neural principles in general, we can go all the way down to C. elegans and these 302 neurons. So, but even when it comes to the cortex, the architecture, the layered structure, the neurons that exist in each of the layers and their functions are pretty well preserved, so that in many ways, and when it comes to particular functions such as space, then the rat and the mouse has actually told us quite a lot. And that's not only because rats and mice are very convenient to work with, and that's a long history of doing that, But there's also that actually navigation and spatial mapping are pretty well developed in rats and mice. So there's a lot that we can learn from them, and for that reason we have still stayed with rats and mice in most of the work that I will tell you about today. So then a little bit of history. So we can't talk about space and spatial maps unless we mention Edvard Tolman. So Tolman, as most of you probably know, worked in the 1930s, 40s, and until mid-late 50s on navigation in rats. So that was part of his work at least, and he proposed that rats and humans have internal maps of the environment, what he called cognitive maps. So those are maps that represent the local space, but also not only the space as such, but also the contents of the space, including the experiences that the animal or the person has at those various locations. So much of Tolman's work was based on experiments with rats, where rats, do you have a pointer, by the way? Or yeah, I forgot, always forget. Or they take it at the airport, one of the two. But what he did was to train rats to do various spatial tasks, and you see one of them here. This is the famous Sunburst Maze, where the rat in the training period walked from start at the bottom and all the way up to a goal through a central area, and then did this over and over again. But then on a test many days later was exposed to the same environment, except that now the last bit through the long arm to the goal was replaced with many arms, one of which was directly to the goal. Oh, thank you very much. Directly to the goal here, and of course what the rat did was to take the direct path. So although this experiment has some conceptual problems or some technical problems with interpretation, that doesn't matter for here, because the important part here is that this led Tolman to suggest that rats are actually able to take shortcuts or detours if needed, instead of just following exactly the route that they were trained. So instead of going up here, which would be the most similar to the training situation, the rat actually chooses the direct path where it has never walked, suggesting that it actually has an internal map of the environment and knows where to go even without not having walked that path before. So this led Tolman to suggest that there are maps in the brain, but he did not want to speculate where these maps are and how they are generated. So like all other behaviorists at that time, he treated the brain like what was often referred to as a black box. That's a big unknown like this. There are inputs coming into the box, there are outputs going out. So the inputs is the stimulation, sensory stimulation, and so on. Outputs is the behavior, and then a psychology or a science of psychology was built up around links between the inputs and the outputs without invoking any neural mechanisms at all. So this made quite a lot of sense at that time, because the technology for really studying neural activity wasn't all that developed at that time, and also concepts were even not present, so it was very, very difficult to approach. But at the end of his life, Tolman actually said that had he then started, he would actually, this was after Hebb had published his work, then he said that he would actually start, he thought now was the time to dig into the neural mechanisms. But then 1950s is when a lot of things changed, so optimism came back. And one of the things that happened was the important development of microelectrodes. And for example, the tungsten electrode, developed in 1955 by David Hubel, made it possible to record activity from individual neurons, and as shown then soon after by Hubel and Wiesel, for example, it was actually possible to pick up the activity of individual cells while animals, cats, monkeys, participated in simple sensory tasks. So that was a major development for neuroscience. And that also changed things for neuroscience of space, which was to come a few decades later. And the important event here was in 1971, when John O'Keefe started to record from single neurons in the hippocampus of really moving rats. So this shows a rat walking around in an environment. So John used different types of environment, but they all had in common that the rat walked freely. It wasn't constrained in any way. And he just listened to the activity of the spikes and saw what behaviors it correlated with. And I think to his and everyone's surprise, there was a strong correlate with the animal's position in space, which I will show you very soon. So this was the basis for the discovery of place cells, and even you may probably have seen this movie before, but I always play it just to bring everyone on board. So this is now one rat walking around in a box that is 1 by 1 meter large. Seen from above, the rat is walking around and picking up chocolate crumbles, which it likes, very small, so they don't interfere with the recording. They just lick them and swallow them. And this is just in order to get the rat to walk around and cover every possible space in every possible direction in the environment. And while the rat is doing this, we are then listening to a single cell in the hippocampus. And what you hear corresponds to the action potentials of that cell, and you also see the location that the cell, where the rat is when the cell is firing. Now we speed up a little bit, but I guess you get the point that this cell is active only when the rat is up in the top left corner, more or less. Otherwise quite silent. And this can be color-coded. Red means high activity, blue means low activity. And this is one example, place cell. Other cells fired in a similar way, but at different locations. And it was then pretty clear soon that among local groups of place cells, there was enough activity to cover every possible space in the environment. And given that it was possible to infer the rat's location from the combined activity of cells anywhere apparently in the hippocampus. So this then led John O'Keefe and Lynn Edel to suggest in 1978 that this might actually be the basis of a neural map of space that was then located among place cells in the hippocampus. Then, now making a big jump till 1996, this is just after Maibrit and I were in London to learn the place cell recording technique and set it up in Trondheim. One of the questions we asked is, where is this signal generated and how is it generated? So in the mid-1990s, and still today this is not well understood, but we know a little bit more. So one of the what we knew in the mid-1990s was that most of the place cell activity had been recorded in the CA1 area of the hippocampus. So this shows a transection through hippocampus, entorhinal cortex here, and then most of the input comes in like this, goes through a loop through CA3, dentate, CA3, CA1, and out. Somewhat simplified, but nonetheless, the one-way traffic is a pretty unique feature of the hippocampus, and place cells had almost exclusively been recorded in CA1, some in CA3, and a few papers in dentate, but very much in CA1. So one of the ideas was, at that time, was that this place cell signal was maybe not generated in hippocampus, but at least amplified in some way in the intrinsic circuitry of the hippocampus before the signal gets to CA1. So an obvious experiment to do at that time was to inactivate or lesion the earlier parts of the circuit and then see if the place cell signal in CA1 disappeared. So we did this together with Vegard Brun in 2002 and found that there is actually still place cell activity even if you disconnect the CA1 entirely from its inputs within the hippocampus. So this shows 7 different cells in the same box that you saw, and you see that they all have spatial activity. They all have preferred firing locations. So that led us to ask then, could there be direct inputs from the entorhinal cortex? We know there are, and would it make sense to actually go to the entorhinal cortex and look for spatial signals there and try to understand how much actually came from outside? And so we did. This is work with Marianne Fyn and a few other students recording in medial entorhinal cortex. This is a rat brain seen from behind, and this is the medial entorhinal cortex. Most of the recordings were done here in the dorsal part, which has strong direct inputs to the part of hippocampus where most of the place cells are located. And this we could do because we work together with Menno Witter, which is an anatomist, an expert on entorhinal anatomy. And this then brought us to find grid cells, which I'll now introduce to you by showing a movie. Our sound is on, so you'll hear it very soon.
This rat has an electrode implant that enables us to listen to the activity of one single grid cell. The electrodes connect directly to the entorhinal cortex of the rat, which is the area of the brain where the grid cells are. The microelectrodes that go into the brain are very thin, only 17 micrometers wide, so small that they can pick up neural spike signals from individual cells. By connecting a cable to the implant, we can record the grid cell activity on a computer. On the screen, we see the neural discharges appearing as colored dots corresponding to the rat's location in the box. We hear the popping sound of the grid cell discharging in real time as the rat is exploring the surface of the box looking for chocolate sprinkles. If we speed up this process, a pattern emerges. The dots indicating neural activity are clustered. Together, they form a hexagonal pattern that tiles the entire surface of the box. The grid cell maps the rat's spatial environment by organizing it into a regular pattern of electrical impulses from the neuron.
Okay, so many of the cells in the entorhinal cortex, in the medial entorhinal cortex, turned out to be grid cells. So there was perhaps the most common cell type, at least among the ones that we have observed so far. And they are particularly dominant in layer 2 of the medial entorhinal cortex. So here you see a rat brain from the side. This is the entorhinal cortex. This is medial entorhinal cortex in red color, and this is dorsal, this is ventral. And it turned out that these cells are organized in some ways and not other ways, but one particular pattern is that as you go from the dorsal part and towards the ventral part, the scale seems to change, so that on average you have a small scale at the dorsal part. You actually only seem to have cells with small grid scales. That means that there's a short distance between each of the fields of the grid up here, and a larger scale as you go down. So the grid pattern of these cells then increases on average as you go from top to bottom. So this is not a continuous change. It actually turns out from experiments that I will not go into because that's published. But it turns out that there are multiple levels, beginning with the smallest scale in the dorsal part here. And as you go down, you add on additional levels with a larger scale, so that the wavelength or the distance between the fields is larger. So you have a second one as you go down here, you get a third one, and then a fourth one, and probably a fifth one, maybe more as you go further down, which we don't know. Because, but they each have discrete scales. So the smallest one has a distance between fields of approximately 30 centimeters, and then the next ones have 40, 50, and then you get up to the largest ones we have is more than a meter, and then it gets difficult to study because the boxes we have to see the repeating pattern aren't large enough. So, so there is a spatial organization of the scale, but in addition, you also have another pattern of organization, or if you want, lack of organization. And that relates to what we call the phase of the grid. So that is how the grids patterns of different cells are related to each other in XY space. So you see here an example of 3 different grid cells recorded at the same time, one in green, one in blue, one in red. And you can see that they have a particular spatial pattern. That means that the red one, for example, is always a little bit to the right of the blue one, and the green one is below the 2. So the pattern is like that. So this pattern is retained as you go from one environment to another. So this is a schematic, but more or less shows what it would look like if you recorded in a different room, for example, with different features. They still would retain that pattern, so that the red one would be to the right of the blue one and the green one below the 2. And then, of course, as you go back, you get the same. So this shows that the pattern, there actually seems to be one single map, more or less, at least within clusters or what we call modules or grid cells. This structure, phase structure, is, first of all, seems to be random-like because you can't predict the position of the grid fields of one cell from the other one. And the other thing is that that pattern, which seems pretty random, is still preserved as you go to all kinds of other environments. So this is what we have known now for quite a while. And now I think it's enough background, and I'll begin to look at some new data. And then please feel free to ask all kinds of questions, because I will just talk until time is over. And so I know a particular end to this talk. But the question first here then is, how general is this map? If there is more or less a single map, or at least one low-dimensional map, as is also often referred to, does it only apply to states when rats actually walk and explore environments like we have done here, or can it actually extend beyond? So one of the experiments we are currently doing is to study relationships between grid cells when rats sleep. So this is the work of Richard Gardner, who has been recording now for a while among large numbers, or at least trying to get large numbers of grid cells at the same time. And then he is asking if the phase relationship or the XY relationship of these cells in the awake state is maintained when they go to sleep. And of course, he can't measure the XY relationship in sleep, but what he can do is to ask if the same temporal relationship is preserved. So what you see here is a recording from an animal that's walking around in the same type of box. It's 2 different cells, and you can see that these 2 example cells, they have more or less the same phase. That means that the peaks are pretty much in the same locations. So these are 2 cells that are in phase. This is just the color version. And as expected, if you cross-correlate these with regard to time, then you get a big peak in the center here at 0 seconds, because if this shows the probability of cell 2 firing if cell 1 fires, and of course if cell 1 fires here, it's very likely that cell 2 also fires, so that's no surprise. But the more interesting observation is that if you do the same cross-correlation in sleep, you also get the peak here. Same thing, so that means that those cells that fire together in the sleep state, in the awake state, also fire together in the sleep state. And conversely, if you take 2 cells that are out of phase, so you see these are pretty much opposite grid patterns, then you have a dip here in the wake state, but that dip is also present in the sleep state. And 2 pairs of cells might not convince you, but if you now take 1267 pairs of cells and then arrange them according to the degree of cross-correlation in the open field or in the awake state here. So each line here is 1 out of 1267 cross-correlograms. So each line is then, it's the same as this, but it's color-coded so that yellow is a high peak here. And then it's arranged around 0, which is the time of simultaneous firing. And then you can see the highest ones here and the ones with the weakest cross-correlation or inverse cross-correlation at the bottom. Now the point is, if you take the same pairs of cells and then investigate them in slow wave sleep, then you see that you're pretty much the same pattern. It's at least largely preserved, so that there's a very high relationship between which cells fire together in the wake state and which fire together in sleep state. And same, yeah. Mm-hmm.
So, in the awake state, the idea is that you drive these grid cells with an external source, which we relate to path integration. So, in the slow wave sleep stage, what's that driver?
Mm-hmm. Now that's an excellent question, because they are not walking, but then what moves it around? So first of all, I mean, the ultimate goal is to have enough cell that we actually can reconstruct the path, not only see cross correlation. So I think from this we can infer that there must be more or less the same grid map, because those cells that are together in one state are together, fire together in the other. But it would be really nice to, I would expect to see that we actually would at least see fragments of paths in a kind of virtual space. But what would then drive it around? Well, I mean, what would be interesting to see is if the speed cells are probably not silent. I mean, they also have variable activity. So perhaps this is just going on randomly. What would drive it? I can't tell yet, but I think the next step on towards answering that question would actually be now to see whether there are fluctuations in the speed cells that could account for any such movements. But to do that, we really need to have more cells.
And it's unique to slow wave sleep, or you also see it in, let's say, a REM phase of sleep?
Good question.
You want to talk about that?
Here's the answer. We think the same way, right? Yeah, no, it's the bottom line here. I won't go into the details, but you can see it looks just more or less the same. It's more noisy in REM sleep simply for the reason, 2 seconds, simply for the reason that there are fewer cells and fewer cell pairs, but it's just the same. Now there was a question.
Yeah, sure. Do you observe phase precession in these cells?
Well, it's hard to study phase precession because there is no theta activity, right? So, I mean, you could probably, I wouldn't know what you would reference it to, if because phase precession is at least normally studied in relation to theta activity, and there's no theta as long as they in the slower sleep. In REM sleep, I think I agree, we could do it, but the data are still, it's too little really so we haven't gone into it. I think we need more sampling. So I think with a larger data set, we could actually investigate that, but we haven't done it, so I can't tell. It's an interesting question. Yep. Well, I mean, depends on how you see it. I think this is pretty independent, actually, of memory. So it also raises questions about what's the relationship between this and so-called replay of place cell activity, which is known to occur at least in the hippocampus and possibly also reflected in entorhinal cortex. What we see here is independent of whether there is any replay-like activity or sharp wave ripples in the hippocampus. So it goes on all the time. So the way I currently see it is that this is just reflects the ongoing activity of the network, regardless even of memories. But I don't have any direct evidence for that.
But you do have a way of seeing if it is locked to the ripples in the hippocampus, right? To see if it is something also that happens in the awake state.
Yeah, there have been papers published that suggest that this activity co-occurs with ripples in the hippocampus, and I think that may certainly be the case. But we have also explicitly compared the occurrence or the cross correlations between events when there are ripples in the hippocampus and where there aren't, and it's totally the same, absolutely the same. So that I still think the last word hasn't been said about relationship to ripples. My view is that this happens regardless of ripples, really, and it's not really dependent on it. There may be ripple activity on top of it, but what I show you here, I think, is independent of ripples. Good. Then how about hippocampus? Just this is known, and this is published, and everyone here, or many of you probably know that hippocampus is totally different, because place cells in hippocampus, they don't carry one pattern of activity over from one environment to the other. They actually have, you can't predict the firing pattern of one cell from the firing pattern of another. So this shows an experiment where, a typical remapping experiment, where place cells are recorded in environment A, environment B, and back in environment A. And then you can see that, for example, for the first cell here, it fires up in that corner. Now it's silent here, and then up in that corner. The second cell fires down here, and up here, and down here. But there is no particular pattern in how one cell fires here. There's also a large subset in each environment that don't fire at all, like this cell, actually much more than indicated by this example here. And you can't really predict from which cells are inactive in one environment, which cells will also be inactive in the other environment. So we investigated this more thoroughly by doing an experiment a few years ago. Charlotte Alme, this is annoying. I think I'll turn off the email, sorry about that. I didn't know I had it on. Let's see. Okay, and then back. So what she did was that she recorded place cells from 11 different rooms. So it's a long sequence of tests where she split recordings on 2 different days. She had one familiar room where the rat was trained for a long time, began with that, and then had a pair of recordings in one novel room here, a second novel room, a third, fourth, fifth, and back in the first, and familiar, and then to the 6th, 7th, 8th, 9th, 10th, and again 6th, and then familiar. So the question was then, with so many rooms, could you see any recurring activity when she recorded from hippocampal cells? And these are CA3 cells. And what she saw is that the correlation between activity in the different rooms is shown here in a color code. So this is the population vector correlation, so all the cells stacked on top of each other, and then compared for all possible pairs of rooms. So the pairs of the rooms are shown here on the x-axis and on the y-axis, and then the color shows for each combination how similar the activity is across the whole place cell population. And of course, if you correlate the same room with itself, you get a high correlation in red. But then otherwise, you can see that it's pretty much blue, with the exception of a few combinations here. And all of those exceptions are instances where the rat actually has been tested a second time in the same room. So for example, N1 here, N1 there, or here it's N6 versus N6. So, but if it's not a repetition, you can see the correlation is very low, suggesting that the hippocampal place cell code is actually almost totally orthogonal across rooms. So there's no similarity, and it's not different from what you would expect by chance. So,
do you have some metric with which you vary the rooms? Like, are they different, like in a morphing experiment different? Are they different with local cues? Or a combination of local global cues?
No, so we actually used, well, you have seen our lab, so then the rooms are so similar that you can't, it's 11 different rooms. They all consist of a box. They're pretty dark, and there's a curtain around. The curtain is open for the occasion, but they are so similar that I can't actually tell which room is which. I have a slide which actually shows 11 rooms, but it's not here. But it's totally hopeless. I can't tell. But the rat can tell, because the rat has a different population code for every possible environment, even if it's only been there once. I think that is pretty amazing, actually. So they are not easy to distinguish, and still the hippocampus creates a new map for each of them, completely new map. So that, I think that tells a little bit about the differences between entorhinal maps and hippocampal maps, because the entorhinal maps are are really rigid, low-dimensional, or just have, at least for each module of grid cells, they have only one expression form, more or less, in all environments, whereas the place cell map is very diverse and seems to create a new combination of new combination of active cells and firing locations for every environment. And that's exactly what you want, probably, if you want the hippocampal code to encode not only metrics of space, but also the content of space, the experiences that take place there. Then you want to have unique maps for unique environments and experiences. In that sense, you may want thousands of them and not just the same map over and over again. Good. Well, then, gentlemen, just a brief intermezzo, just to again bring everyone on board. There are not only grid cells in the entorhinal cortex, there are also other cells. Head direction cells have been known since the mid-1980s when they were discovered by Jim Rank. So these exist also in the medial entorhinal cortex among the place cells, more in deeper layers and intermediate layers than the grid cells. These are cells that do not have a spatial firing pattern, necessarily, but they have a strong directional firing pattern, which is shown in those polar plots here. So this shows the firing rate of this example cell here according to which direction the rat's head is pointing. So when the rat's head is pointing to the left, as you can see here, the firing rate is high. No firing when it points down or to the right or up. And this is the same on two different locations. And this cell shows the same when for a different cell, which fires when the rat is walking from bottom right towards top left. And again, it's scattered pretty much over the entire environment. Some of these cells are actually grid cells at the same time. So they fire in a grid pattern, but only when the rat is walking in a certain direction. Then there are also border cells. These are cells that fire specifically along certain borders or walls of the recording environment. So in this case, and along the east wall here, and that happens regardless of the shape of the box. If you stretch it in this direction, still keeps the firing along the east wall. And if you stretch it in the other direction, it still keeps it here. Take it to a new room, chooses a new wall, the west wall, and you insert the wall here, it also fires along that wall. So these are recorded in the medial entorhinal cortex, but pretty similar, maybe not identical type of cell was found by the London group, including Neil. Where are you, Neil? Are you here? There you are. At about the same time in the subiculum, but they have some differences where one difference is, and that's why they call their boundary vector cells originally, at least, because they not only fire along the walls, but also at certain distances from the walls. And then the third type of cell I wanted to tell you about is cells that fire strongly in relation to the speed, to the running speed of the animal. So some examples are shown here. And these are 7 different cells, each shown in a different color. So this is time over 2 minutes. And in gray in the background, you see the running speed of the rat. And then the firing rate is shown in color. And what you can see, for example, if you look at the yellow one here, is how closely the firing rate actually follows the running speed of the rat. And that's independently of firing location. So for these 12 different cells shown here, you can see from the color that the cell is pretty much firing everywhere, but still has a linear firing rate relative to the speed of the animal. So then, that also relates to the question about what happens during sleep, and of course one of the things we would want to know is whether there are fluctuations. What is the dynamic range of fluctuations of these cells when the rat is actually sleeping? But they are thought then to be an important part of what actually drives the grid map to be updated according to how the rat or mouse or whatever moves around in space. So what I want to do now is to focus a little bit on slightly more complex environments, because the environments that we study in our labs and also in most other labs are actually very, very simplified. So, for example, they are all open, they're all empty, and they're all small. So what happens if we try to extend the complexity of the environments a little bit? So,
before we go there, just for clarification, can you say something about the relative presence now of border cells, grid cells, speed cells, and so on, and how it maps to this idea of modular organization?
Well, first the relative, the numbers of them, it's hard to say really because we don't have enough cells, but still, I mean, grid cells are, if you record in layer 2, I would say grid cells are probably the most abundant ones. But if you record in layer 2, you will at least have 10% of border cells, so you'll at least have head direction cells are very abundant in the deeper layers, and the deeper you go, the more of them you see. And then there's another type of cell, which I'll present in the next section, which also is about 10% of the cells. So they are mixed, and you can't distinguish them just by location. So they probably interact very, very closely. Then regarding modules, the only thing that is really modular is the grid cells, as far as we know. Then the most abundant modules is the small-scale ones, the one with the short wavelengths, and then it gets less and less as you go to the higher modules. You almost, in a recording of 100 cells, may just have 1 or 2 or 3. Good. So how about the more complex environments? This is work of Tania, who is just completing her PhD in our group. But before I go to her work, just a little bit of background, because I want to step a few years back. That was at the time when Dori Dörrikman was a postdoc with us. So the question he asked was if there is always a continuous map, or whether that continuity is sometimes broken. So what he did was to record from a square environment, like the one here you saw, and this is just a curtain around it, a circular curtain. And then he saw a grid pattern, as expected. But then he converted this open space to what we call a hairpin maze. So this is just a long series of walls, which then forced the rat to go back and forth, back and forth, back and forth, and back and forth until it got to the end here. So starting at one end and going through these hairpins until it came to the other end. And the question was then whether there was a master grid that is overlaid on these hairpins as well, or whether there's a different pattern when you break up the environment or when you structure the environment in this way. And this is what he saw. So first of all, you see the typical expected grid pattern in the open space. But when the rat has to walk in these hairpins, then what you can see that there is no grid pattern really lying on top of it. You see it, for example, here. Instead, you see that in each alley, it's the same pattern, starting over and over again, so that you have the peak at the same distance from bottom to top or top to bottom, regardless. And this happened to almost every cell, so that it seemed like the grid pattern was actually broken at each turn of the maze, and then the rat walked a certain distance, and then it fired, and then it walked, and then it fired again. So seemingly somewhat repetitively from one end to the other, but in any case, there was a sharp break in the population pattern each time the rat turned. So that suggested to us that perhaps grids there's not a universal grid that covers the whole world, but they cover small fragments of space, and then at salient landmarks or whatever, then it's all reset and begins again. So if that's true, then the grid map of the real world actually consists of thousands of small grids that maybe just extend for a few meters, and then they are somehow put together in a way that we don't quite understand. So there are also other indications suggesting the same. So even if you record from large open fields, similar to those I show, if the field is quite large, like this one is 220 by 220 centimeters, you can actually see that there isn't a homogeneous grid pattern all over. So the orientation here, for example, is different in one end and in the other, suggesting that there are effects from the walls or the local effects that don't apply all over. And if you analyze this more thoroughly, you can actually see that you can predict the shifts of these distortions, and you can describe them pretty much as an effect of what is often in continuum mechanics referred to as the shearing forces, where you have strong forces near the walls, and then those forces decrease as you go away from the walls, and you get effects along the walls that you can break down the environment into small fragments like this. You have, for example, parts of 1/9 of the environment. You can then investigate the grid pattern in each of them. And what you find is that our strongest distortions of the grid pattern are actually present if you are close to the walls, and particularly near the corners of the environment. And as you go towards the middle, you can often see a pretty sharp break in the grid pattern in the middle, which then suggests that, like in this hairpin maze, there are actually mini-grids that are somehow glued together in the center. So we wanted to understand that better, and then that brings me to Tania's experiment, which is the ongoing work, where what she did was to train rats to walk in a simple environment, but now different from the one you saw, in the sense that there is a wall that separates two halves of the environment. So the whole thing is a 2 by 2 meter box, but then in the middle there is this wall that breaks it down into two rectangles of 2 by 1 meter each. And what she then does, after a while in training the rats here, is she takes away that wall and then asks, if you have a grid map here, you have a grid map here, how are they integrated once it becomes one environment? Do they somehow fuse together, or do you retain two different maps? So you might have the possibility that maybe one of the maps wins and just extends from one end, from the A end, for example, into the B end. You keep a continuous map. Another one is that you actually somehow get a compromise in the middle, that they are integrated. And the third one is that you get a completely new map that is not similar to any of the original ones. So there are several possible outcomes, but what she found is that the compromise solution seems to be the one that takes place all the time. So what she did was that she recorded grids, as I said, in environment A and environment B, and then afterwards in what we call AB, which was the open environment. And then she used the sliding correlation analysis, which just means that she took small fragments of space from one across the entire environment, and that took the local correlation, spatial correlation, in the original environment, and then compared with the same area of the combined environment, and then color-coded that local correlation. So red means high correlation, and blue means low correlation. If you do that for single trials, it's very noisy, but if you do it for all the trials of the rat, it starts to get better. And if you combine it for all the rats, you start to see the pattern. And here you see the pattern, which is then that if you now remember that the wall is horizontal here in the middle, so that means that there's low correlation between the original pattern and the final pattern in the area where the wall was. But at the peripheral walls, far away from the wall, the correlation is very, very high, which means that the rat actually then keeps the original grid pattern along those peripheral walls, but somehow creates a new pattern here in the center. So it's not a global response, it's actually a response that takes place locally. So then
So how much experience did they have of the AB?
Quite a lot. I mean, at least a week of training or so. So I have been there many times.
So you know with the paper on this?
Yeah, the carpenter. Yes.
He heroically recorded up to 21 days, and it looked like it was slowly becoming more and more global.
Yeah.
Starting at the bit where they could walk between the environments, but slowly spreading. So.
Yeah, no, I know about that paper, and it's strange. I think probably there are two different ways it may go. Sometimes you may get a global map, which in that experiment happened, and here it clearly never happened. So even if in some experiments Tania could follow even the same cells for many days, and that integration never happened. So it was always this pattern which you see at the bottom. So why those experiments are different, I think that's really hard to say. I would almost expect the opposite, that because in this experiment it is open, the space is open, and I would kind of expect that if there is a global integration of the whole map, that might happen there. Whereas in that other study, then there's a corridor which links the two, so that I never see the two environments at the same time. And then I would more expect that it would take a long time for them to integrate. But what this suggests is that under these conditions, you have the two original maps. They keep their anchoring or the association with the external world in the periphery here, but then they make a compromise in the middle. And I'll show you what we actually think happens here, because yeah.
For clarification, Tony also has a question there.
Oh, sorry, I didn't see. Oh, yeah.
Are border cells and grid cells a very distinct population? Because some of these results could be explained if grid cells morph into border cells when they're near to the borders.
Well, I think partly because we did record some border cells, and there is a change in the border cells, at least obviously, in the sense that those border cells that had fired along the central wall, which wasn't there any longer. Of course, they did not continue to fire in the center. They then fired only along the peripheral walls instead. So there is a reorganization in the center, which I probably think has consequences or is part of the dynamic mechanism that goes on in the center when you do this. But yeah, I think it will become clear from the next 2-3 slides what may be going on.
So before we go there, what's interesting is that the minimum correlation there is 0.4.
Yeah.
And the max is 0.8. So you could also argue that what happens in the center is just a merging of the two maps.
Yeah, sure. That's exactly what I'm going to show. no, that is.
Sorry.
No, that's it's good. Then I know that you are you are with me so far. No, that's the question. Is it a merging of the two maps? And one way to begin to study that is to try to identify the fields of the grid pattern, which you see up here. And then if you use a field detection algorithm, the black dots here are the detected fields. So this is again in the original pair of environments, and this is in the final pair of environments. And what Tania then did was that she asked for each field, what is the distance to the nearest field within the range that corresponds to, I think it was half of the grid spacing or something like that. But then she gets a lot of vectors and then plots all those vectors for that single cell on top of each other. And if you then measure the lengths of these movements or these vectors, what you see, if you color code the length of the vectors, this is what you get. You see that the movements are the largest in the center again, so that is red. And then blue means little movement. So you see in the periphery here, the fields, as expected, don't move much because it's so highly correlated. But what you get here, you still have the fields, but they actually are in different positions. And then you can do this in a way that also is independent of detecting fields, because we could say there was something suspect with the field detection mechanism. So we also did it in a cross-correlation way, which just took chunks of the environment and then cross-correlated with the same chunk of every possible part of the environment here, and then did it for the entire space. And again, what you get, this is for all cells of that rat, and for all cells of all rats. Again, you see that red color means that there's the biggest shift in the peak of the local cross-correlation window is in the center, and blue then means that the peak of the two cross-correlation analysis is pretty much in the same place. So that's just consistent with the field analysis, which suggests that the biggest reorganization or the biggest movement of fields is in the center. But then, does that mean I mean, what we really wonder is this just a reorganization towards towards getting a local grid pattern? But because you can imagine that before you do the fusion, and when you have the wall there, in many of the cells, these two maps are not in phase. That means that if you just removed the wall and the cells kept its original firing pattern on both sides, you will have sometimes a very sharp break in the middle where you don't have continuity in the grid. So ideally, what we would want to do is to establish for every possible place in the environment the local grid pattern. That, though, in order to say if there is to measure the symmetry of the grid pattern at every possible place, it's a bit difficult because you need some space simply to tell if there is a local grid pattern. And for that, the environment was a little bit on the low side. So what we did instead was that we just measured the distance to all neighboring fields, not requiring that we had 6 of them around, and then measure the variation in the distances to neighboring fields for every possible field that we detected, either before removal of the wall or after. And then the variation or the standard deviation in those distances to neighboring fields was then color-coded, and this is before wall removal. Then you see, as expected, at the center, there's a high variation because the two parts of the grid here are not necessarily in phase, so sometimes you really have a large variation in the distances to neighboring fields. But afterwards, there is not a larger variation anymore along the center here than in the rest of the grid. So then it is, you actually see the largest variations in the periphery often, which reflects these effects of the walls that I briefly showed afterwards. So this is consistent with the idea that there is actually a fusion of the two grid maps where they keep their anchoring to the peripheral parts, and then, as Neil said, that may or may not happen, but in our circumstances it does happen. And when they keep their peripheral anchoring and then they are most often out of phase, then what happens in the center is that the grid fields reorganize themselves so that everywhere in the environment you get a locally symmetric map that is, there's no breaks in the map really, which probably is what you would like to have if these maps are somehow used for actually helping you to navigate from A to B, because then you don't want to have sudden discontinuities in the grid pattern. Yep. So first there and then there. Yep.
So question is regarding speed cell. So question is regarding speed cells. And because whatever you have a border, what you have in a slowing, the rat cannot move from there. And actually, any movement starts from the wall. Mm. So if you previously showed that during REM you retain the temporal relationship, this means that you just have an internal pattern that you anchor to the space. Whatever, and the movement of the animal in the space is what determines how they are anchored to the space. So what are these speed cells doing when you split the...
well, yeah, I think what I think happens is that, I mean, this shows that there is a strong anchoring of the grid map to something in the external environment, and that happens in the periphery here. But then in addition, I think the animal uses what we call path integration, so uses its own self-motion, which probably relies on speed cells in order to calculate position relative to some reference point, which might be the walls. And then I think that helps the animal or this map to keep some continuity from one end to the other. And when the wall here is removed, that continuity can then persist from one end towards the other into that other environment. But then the same happens from the other side, and those continuities don't really then mean that in the center you don't necessarily get the firing in the same place. So somehow the whole network seems to reorganize itself to become more symmetrical than it would be if it just moved the self-movement. But what exactly happens in the speed cells, we haven't measured from the speed cells, but I think it is related to path integration, but I can't explain it all. There's something with the network reorganizing itself.
So do you have any intuition of what's happening with place fields at the center location of this maze?
What did you say?
So if you have an intuition of what's happening with the place fields for cells that are representing the...
Oh, yeah, the place fields in the same task?
Right.
The center.
But we haven't recorded from place cells in this task. So that I don't know. But I would expect that something similar would happen. So I was yesterday actually in Hungary. So and John O'Keefe was there too, who presented data. You may know this, Neil, but I mean they also recorded from place cells in a somewhat similar task, where I think what they did was that they had these strange environments, trapezoids and so on, which they shrinked or changed, and then also saw that fields moved along with the walls. And he showed briefly that something like that also happens in place cells. So I think the two maps are more or less coherent in that respect. But I don't have data from the hippocampus to show.
Would you see a causal relationship in between them? Would you see a causal relationship between them? So, I mean, we remember that you had like a projection from C1 back to Entorhinal, and you see disruption of grid cells, right? So, would that be an effect of remapping of place cells being affected on the grid network?
Yeah, no, again, what he showed, but very briefly, and I can't really tell, is that under these circumstances there was no remapping. But I think based on work that have done before, if the changes are small enough, you don't get remapping, then you just get movements.
So we did a lot of experiments in the 90s and 2000s on place cells and this kind of thing by inserting or removing barriers.
Yeah.
And the typically a place cell, if it fired, say, against the south wall of the box, if you put a barrier in, it will also fire just above the horizontal barrier, which is why we predicted these boundary-related inputs, which subsequently we've all found. And so in this experiment, if you have a place field, and we did something similar, that fires in the middle with a barrier there, then you remove the barrier, it dropped down to the nearest boundary below it, if it's firing above it, or if it's firing below the barrier. So to some extent, and over a small number of trials, things change if you record from a cell for a long period, but for place cells, they seem a little bit perceptually driven, sensory driven by inputs from boundaries. And if you take away the boundary, it will find the next boundary in that direction to attach to.
Yeah.
So Edvard, this is pretty sophisticated analysis, but in some sense the results are a bit worrisome, right? Because you see that you have a similar change close to the walls.
Yeah.
Well, the walls are supposed to anchor.
Yeah, but then with the walls, they anchor, yes, but what you also see with the walls is what I showed just very briefly here, where, I mean, the walls also cause distortions of the grid patterns, and I think that's actually why, if you have an open space, we over and over again see that the most regular grid pattern is if you go away from the walls in the center, and as you get very close to this, especially the corners, you get much more distortions. Okay. One may also wonder how fast this develops, and in fact it is pretty fast. It's almost expressed as soon as you can record it. So we try to break this down into small fragments, but of course, since we need a certain coverage of space to tell this, we can't really break it down to anything below at least 1 minute fragments. But if we break it into the first 5 minutes, first 10 minutes, first 20 minutes or so, we see that the pattern is pretty much stable from the beginning, the first time they are in the whole open space. And then this is just correlations between the instantaneous firing pattern and the final firing pattern in the open environment. And you can see that between the first and fifth minute there is development, but then after 5 minutes it's pretty much stable. And we also, as I said, followed it for many days in some of the rats, and still no change. So one of the things one may wonder about is, in this environment, what's the relationship between different cells? Because if if this information that is in the grid cells were to be used for rats or for the brain to actually infer location, if the grid map somehow express some metrics that the system is somehow using, then it would be hard to use that information if different cells were squeezed or changed in different ways. It is much easier if the whole system actually changed in a coherent way. And that is what is the case, because for cells that were part of the same module recorded simultaneously, what Tania did was that she recorded the relationship between the grid map in one part, in the A part of the environment, and in the B part of the environment, and cross-correlated the two, and then got the characteristic shift in the peaks of the cross-correlogram. Start over again. What she actually did is that she correlated the A part of the map for a particular cell with the A part of the map for a different cell, and then she saw that those two maps of those two different cells were actually shifted, as you would expect, because you have all kinds of phase relationships. So in this case, the C7 cell is shifted a little bit upward to the right compared to the C3 cell. And if there is such a shift in the A part of the environment, that shift was also present in the B part of the environment. And if you now remove the wall, she found that that shift was still present in both the A and the B parts of the environment. And importantly, if she focused the analysis just to the center, it was still that same shift. And this just shows quantification, shows the difference in the distance of the phase shift, and then compared between the real data and some random data where she randomly selected a different cell and compared with that. And you see that it's very different from what you get from a random combination of cell pairs. So the shift is the difference in the amount of shift in the preconditioning here and in the post-condition is very, very small. Just 2 bins, that means 4 centimeters. So it's really, really small, which then suggests that probably the whole map or the whole module of grid cells is retained in the same way, so that if the cells now in the center reorganize so that fields are stretched in one way or the other, it's more or less the same way in all of the cells and not just in one of the cells, which then probably suggests, although that hasn't maybe clearly been shown yet, that you can actually infer location to some extent after all, even if there are these local distortions. So that now brings me to a second part of the... So how am I with time, actually?
Well, we have another 15 minutes or so, 10, 15 minutes.
Okay, so I wanted to address 2 things, but I'll try to get into both of them. They are much shorter. But the first one is still about complexity of environments, because most environments contain objects. They're not just open spaces. So what happens if you put objects into the environment? This is the work of Øyvind Højdal, who is a PhD student in the lab. And what he did was that he put objects into the environment. So this is a Lego tower that is put into the box, and otherwise the recording conditions are just like they are in the experiments I've told you about. But what he found was that there is a pretty substantial fraction of cells in the medial entorhinal cortex intermingled between the grid cells and the border cells that actually fire in relation to the object. And those cells don't fire at the object as such, but they fire at a certain distance and direction from the object. And for that reason, we now refer to them as object vector cells, because you can define their position as a vector in relation to the position of the object. And for example, with regard to orientation of the object, he divided the map into these radial bins and then sees that there is a high firing rate when the rat was on the west side of the object, but then no firing when it was on the east side of the object, and then again when it comes back to the west side, it's high firing. And it's also tuned to distance, so that in this case, the maximum firing is about, hard to say, 5 to 10 centimeters away from the object. And there are many such cells, just some other examples shown here. Some of them have a single firing field, like this one firing on the north side of the object, maybe 15, 20 centimeters away, and same again on a different trial with the same object, object slightly displaced now, but still on the north side. Some cells fire on two sides of the object, like this one, and this is not a consequence of poor separation of the cells, because we were concerned about that, but some of them are very well isolated. So sometimes they actually have two fields, although the common pattern is to have a single field. Here's the one with two again, and here's another one with one. And then the distribution of these fields is shown here. So this shows each line, each row here is one cell, and then color is firing rate, and this is the direction relative to the object. And you can see that all directions relative to the object are represented. That means that you are firing around the whole object, but still you can see it's not totally a straight line. There's actually some preference for some orientations, and those orientations correspond to the walls of the box, so that there are more cells that have an orientation that is at 0, 90, and 180 degrees. It's a bit hard to see, but you can perhaps also see it here, that there are peaks at minus 90, 0, 90, and 180. But only weakly, but still. And then this just shows the distribution of distances from the object. So the most frequent distance is about 10 centimeters, 5 to 10 centimeters. But you have distances that even reach up to 45 centimeters away from the object. And this is regardless of where the object is. So you can put the object anywhere in the environment, and it will still be 45 centimeters on the north side, for example. And this shows other examples. So this shows also that it's independent of the actual nature of the objects. You can have different types of objects. I mean, they are somewhat similar in the three-dimensional shape, but still also quite distinct. And the cell will fire on a certain side, on a certain distance away from the object, regardless of what it looks like, and it will do the same for all of them. So, for example, here there are two objects, and you see that the cell fires to the certain distance to the left for both of them. On this one, it fires on the southeast side of all three objects. On this one, particularly this one, I like, because here we have 1, 2, 3, 4, 5, 6 objects, and you can see that you get a field on the northwest side of every one of the objects. And this one too, you have on the northwest side. So it is independent of the actual content or the nature of the object, but the vectorial relationship to that object, regardless of where in the local space, is the same all the time. Yep.
Are they in the same region where you detect grid cells, or whether they are more in the lateral entorhinal cortex?
No. Yeah, no, I'll come to that, but this is in the medial entorhinal cortex, and they're mixed with the grid cells. So you may have grid cells and object vector cells on the same tetrodes quite often.
So are you going to revisit your border cells? So some people record boundary vector cells in medial entorhinal cortex, which obviously look like this but respond to more extended objects.
Yeah, no, I think that's an important question. That's not totally clarified, but I think what we can say is that none of these cells really have typical border fields, in the sense that they also fire along any of the walls. So they are distinct from them. It's not quantified yet, but I think out of these cells, I haven't seen a single one that also has a field along any of the wall.
That wasn't really the question. The question was whether there's the analogous cells to the object vector cells in the entorhinal cortex amongst the population that do respond to borders.
You mean among the borders,
displaced?
the cells that, if you go the other way, that they take the cells that respond to borders, whether they also respond to an object? Is that the question?
No. My question was, among border responding cells, are there any that respond at a little bit of an offset? I.e. a boundary vector cell?
No. Not among the borders. The border cells, as we have seen, I mean, with the exception of maybe 2 or 3 cells that I've ever seen in my whole life, which actually fire at an offset among the border cells. That is, I would say, extremely uncommon. With these cells, it's different because, as you saw, the distribution can be anything really within the available space.
And do they relate to Jim Neerim's object vector cell?
Good question. Comes in 2 slides. Yes.
In the border cells, you did see the shearing effect.
Yeah.
So that might be a variation on the same bias that you see in these cells, possibly. No? Because now also the visual features you're exposed to might be biasing the overall neural dynamics. So it's not necessarily qualitatively different.
Well, I'm not sure I get quite where you want me to go.
Well, like in this case, there's this offset, right? Also, Neil's asking about.
Yeah.
And Neil was asking, do you see an offset also with your border cells? You say no.
No, yeah.
But you do see a spatial bias in some border cells, which you call shearing.
Well, but the bias that we see is not really in the border cells itself. I'm not sure whether the bias is in the grid cells where the pattern is distorted. The border cells, for the border cells themselves, we don't really see anything that is changed.
But now, if you go one back, then you see that the direction seems to be discretized. Is that an experimental effect, or you think that's really there in the response of these neurons? Is there discretization of this directional tuning?
I think it reflects the fact that in many of these environments, the rat is in a square environment, so that there is an over-representation of directions that correspond to the direction of the walls, whatever that means. Good. Well, just quickly and not surprisingly, if you take away the object, the pattern deteriorates, but it doesn't totally disappear. So there is still some remnant of spatial activity, even if you remove the wall, even if you remove the object, and that even is the case even before the object has ever been there. Of course, then it's not necessarily at the same place, but these cells aren't completely non-spatial. They have some stability, but of course that stability becomes much clearer when there is an object. So then, like the other cells in medial entorhinal cortex, they also maintain their relationships with each other. So some example is shown here. So you have 2 object vector cells here. So this one fires on the southeast side of the object, and this one fires on the northeast side. So they are offset by about 90 degrees. And then here you also have a head direction cell that fires on the southwest side. So if you now take these cells from one room and then record in another room, then this one flips about 180 degrees. So it is now on the northwest side. This one also flips about 180 degrees. So now it's on the southwest side. And also the head direction cells flips 180 degrees. So you can see that the intrinsic relationship here is more or less preserved as you go from one environment to the other, just like all the other types of spatial cells and the head direction cells that keep their internal relationships, suggesting that this is just like border cells and grid cells and all the other head direction cells, part of a single or at least very coherent map that has the same metrics in different environments. And then to the question, how do these cells actually distinguish themselves from other cells? So let's first ask whether, are these allocentric cells? Yes, they are. That means that they fire at their locations independently of how the rat actually moves, or the mouse in this case moves in that environment. It's not egocentric, doesn't depend on whether the mouse goes from left to right or from right to left. So this just shows different directions of movement, and you can see that the cell fires in the same locations relative to the object, regardless of the direction of movement. Same here. You can quantify this. This shows firing rate as a function of movement direction, so not orientation to the object, but the direction that the rat moves. Most of the cells are like this. There's no preference at all. And the most extreme cell is like this, so it fires a little bit more to some movement directions to others. But then if you take the population as a whole and quantify the difference between the highest here and the average, you find absolutely no difference from a shuffle distribution. So they are independent of direction towards objects or towards goals. So in that sense, they're different from what is called goal orientation cells, which have been reported in the one from Nahum Ulanovsky's group in the C1 recently. But then the question about how are these different from the object cells that have been described especially by Knierim's group in the lateral entorhinal cortex. And what Jim Knierim found some years ago was that there are cells in the lateral entorhinal cortex that fire specifically to the presence of objects. So this shows examples, 4 different objects here. And what you can see is that these cells fire around the object, at the object where they are. Sometimes they even, as we have shown in our own work, sometimes these cells actually even continue to fire at the location of the object when the object has been removed, so keep some trace activity. And that can even be kept under some conditions for several weeks. But what is different is that in these cells, it's always exactly at the location of the object, whereas in the object vector cells, it is always, virtually always, some displacement in a certain direction. Yeah. Yeah, that's the next slide. No, that's a good point. Well, exactly. So when we now get to the 2013 paper of Jim Knierim, then there is a population of cells in the hippocampus that is different and much more like the ones in the medial entorhinal cortex. So this study, it had, unfortunately, we don't know how many cells there are of this, because in that paper, I think there were 8 cells that had these properties. But those properties are somewhat similar to the ones in the medial entorhinal cortex, which you see here. It's 4 objects again, and you can see that they have firing fields displaced from the location of the object. So in this side is on the southeast side, on 2 of them. On 2 of the other objects, they don't fire, and here's another one that fires again on the southeast side of 2 of the objects. So I think it's hard to say because, as I said again, there's a very, very low number of cells in that study. But based on the examples that they show, there are at least 2 major differences. So one is that it's only a subset of objects that elicit such activity, as you see here. Another one is that it often was reported to develop over multiple sessions. So it takes a long time for many of these fields to appear in the hippocampus, whereas the ones in the entorhinal cortex are present immediately. So I think those are the main differences. The exact relationship between the hippocampal ones and the entorhinal ones still has to be worked out, and probably should have simultaneous recordings from entorhinal cortex and hippocampus. But I think for now, I think at least the 3 things we can say, that there are many more in the entorhinal cortex. We just didn't see them before because we didn't use to put objects into the environment. And the other is that they are instantaneous, they develop immediately as you put an object in. And the third is that in the entorhinal cortex, in the medial entorhinal cortex, they are present regardless of the nature of the object. As long as it's some sort of obstacle, some sort of thing in the environment, there will be a field at a certain distance from it. And in any case, this is a distinct group of cells. We also investigated the overlap with other types of cells. And in very rare occasions, there are actually some mixtures of phenotypes. And this shows one example where this is a grid cell that actually gets a field at or near the object. So the object is here in addition to the original grid patterns. You see the new field comes in here at the side of this object and then disappears as the object is removed again here. And here also, this is a bigger grid. Then if you introduce the object, you get another field here. And if you remove it, then it gradually disappears again. So there are some grid cells that actually have a similar property in the sense that they add fields near the objects, but by and large, most of these object vector cells are distinct cells. They're different from head direction cells. They seem to be different from border cells, and they are different from speed cells. So, of course, these cells may have mixed phenotypes, and you may have conjunctive types of coding, but it shows that regardless, in this circuit of medial entorhinal cortex, not only does the circuit encode position in an absolute open space, but it also takes into account the metric relationships to the local environments. And those local features may not only be the boundaries, which was proposed by Niels' work already actually before grid cells came, but it may actually also include discrete objects, more punctuate objects, where you actually can then, I'm sure you can think of possible ways that it can actually work much in the same way as originally proposed for the boundary vector cells. So then questions still.
Yeah, well, linked to this, it seems like we're moving from a scheme of spatial metric coding into more the structure of the environment, or maybe even guidelines how to navigate that is coded by Medial and Entorhinal.
Well, I would still say this is maybe a lot of metrics in this. It's just that it is more complicated because it also seems to take into account discrete objects that are in the environment. But the fact that these cells fire at in such a replicable manner at certain distances, certain directions from discrete objects suggests that it is complicated, but still so reliable that I think it actually is using the metrics, and particularly the fact that the cells don't care about the identity of the objects. And that it can happen anywhere, regardless of where the object is in the space. So,
Do we have other questions?
Can you talk about the sensory input into these processes?
Well, what we have done is I showed very briefly the darkness experiment where we remove the visual inputs, and then you still, you lose a lot if you remove vision. So vision is important for these cells. As you can see here, this is an example here, then we'll take that one. It's pretty different as you remove, well, actually, sorry, this is a different experiment. This is where we remove the object. But we also have another one where we just turn off the light but keep the object there. But it still also gets very fuzzy. So visual input is important, but of course that's not all. I would imagine that that's combined with, just like for the other cells, that you use self-motion to actually calculate the position relative to the objects as well.
So Edvard, we're going for questions now, or do you want to conclude with?
Yeah, if you tell me how much time more, 1 minute, 2 minutes, 5 minutes.
Sure enough, 5, whatever you need
Okay.
to finish up. But
yeah,
no problem.
well, let me then just take the final bit. I'll try to do it quickly, and then we can take general stuff, because there is one study that I wanted to highlight, even if I don't have much time for it. And that is the question about the network, and particularly GABAergic interneurons. We know that the grid cells and also the other principal cells of the medial entorhinal cortex are strongly interconnected by networks of interneurons. But then we also, this is the work of Shenglin Miao and Kitchen Cow. And what they did is that they focused on 2 major types of interneurons. So in the cortex, there's a wide diversity of interneurons, but we can still classify them somehow. And now it's turned out during the last decade that 5-10 years that there are actually 3 types that are pretty much non-overlapping, 3 classes of interneurons in the cortex. Those that express parvalbumin, those that express somatostatin, and those that express 5-HT3A. Most of the work has been done on parvalbumin and somatostatin expressing interneurons, and we don't really need to look too much in detail on these stains of parvalbumin or somatostatin interneurons. It's very hard to see here, really. But what we did is to count them, and and ask whether are there any cells that show overlap, expressing both of them. And the bottom line is that there's almost no overlap of them, about 1%, which is consistent, which has been shown in other cortices. So those are really two distinct types of interneurons. Then what we did, or what Kitchen and Miao did, was that they tried to inactivate selectively either the parvalbumin group of GABAergic interneurons or the somatostatin group of interneurons. And the way to do this is that they used 2 lines of transgenic mice. One line, which was PV or parvalbumin Cree transgenic mice, so that they could use that mouse line and then inject into the medial entorhinal cortex an AV virus that carried the gene for HM4D, which is a dread receptor, an inhibitory dread receptor. So this is an artificial receptor that then, when injected into these transgenic mice, causes the expression of that artificial receptor, HM4D, in parvalbumin expressing interneurons only. So you put an artificial receptor into the entorhinal parvalbumin interneurons. Then what you do is to apply ligand CNO systemically, and that CNO targets only and selectively this artificial receptor, so that is in essence what happens is that you turn off the parvalbumin expressing interneurons in that mouse line. And then the same experiment they did for the somatostatin Cree mice, so that they could specifically turn off the somatostatin interneurons in those mice. And this just shows when you first turn off the parvalbumin interneurons, then maybe not surprisingly, that interferes with the grid pattern. It doesn't disappear entirely. That could also reflect the rather low dose, if you like, that we used, but nonetheless clearly interferes with the grid pattern in these cells, and this is reflected in the symmetry scores for the grid cells. And the same happens with the speed cells. They are also much disrupted. The speed score, speed rate relationship is disrupted. So those two are impaired when you turn off the parvalbumin interneurons. But if you then do this with the somatostatin interneurons, turn off them instead, what happens is that there's absolutely no effect on the grid cells. If anything, they just get sharper because the firing rates increase in the fields. But instead, what happens is that there's another class of cell, which I haven't really talked about because until recently we just considered them as garbage. Those are the cells that have spatial firing fields, somewhat like place cells in the hippocampus, not periodic, so they're not hexagonal, but they fire in some places reliably from one trial to another. Quite often the fields are pretty extended, blurred, don't really look nice, but still they're reliable. What happens to those cells is that if you block the somatostatin interneurons, then they lose their spatial selectivity, and then it comes back when the drug is out of the body again. And this happens over and over again in all of those cells. But in those animals, when you turn off the somatostatin cells, there's absolutely no effect on the grid cells. And if you now take the parvalbumin group again, go back to the parvalbumin group and ask what happens to these aperiodic spatial cells, there's absolutely no effect. So what this shows is that if you turn off the parvalbumin interneurons, you interfere with the grid cells and the speed cells, but there's no effect on these aperiodic spatial cells. If you interfere with the somatostatin interneurons, there's no effect on the parvalbumin, there's no effect on the grid cells, no effect on the speed cells, but there is an effect on these spatial cells that don't have a hexagonal pattern. And this is just further examples. So you see in the middle, there's they get much more blurred than they are. But this is the quantification, but skip that. But then to the conclusion slide, this is a double dissociation, because if you turn off the parvalbumin cells that target the soma and are thought to be important for the grid pattern, then you lose grid pattern, but there's no effect on these aperiodic cells. But if you now do the same with the somatostatin interneurons, turn off those, these are dendrite targeting, much slower, so not necessarily good for grid cells. But then there's absolutely no effect for grid cells, but there is an effect on the aperiodic cells. So it's two different classes of interneurons seeming to rely on two different networks of interneurons in the medial entorhinal cortex, suggesting then, at least somewhat to my surprise, that these interneural networks are much more independent than I thought, at least in the entorhinal cortex. So I think with that, I'll skip all the development stuff and just go to the final conclusions, the acknowledgement slide. And that brings me here. So that's another grid pattern. This is the people in our group, and many people involved. Too many to mention. I mentioned those that are particularly involved in the new unpublished studies, but otherwise, original work on grid cells, speed cells, border cells include the people at the top there. First of all, May-Britt Moser, but then also Marianne Fien, Torkel Hafting, Sturla Molden, Francesca Sargolini, Trygve Solstad, Emilio Kropf, Sten Solar for the borders, for the modules, Charlotte Alme for hippocampal work, and several collaborators, important, Alessandro Treves, Bruce McNaughton, Carl Barnes, and Menno Witter. And the others are people whom I already mentioned, and a lot of people pay for this stuff. So I think with that, time to conclude, and any remaining questions, I'm happy to take.
Okay, fantastic. Thank you, Edward. Do we have more questions?
Hi. So grid cells are thought to provide the metric for place cells. So I don't know about rats, but for humans, it's clear that we can recognize places based on vision only also.
Yes.
So when we do that, when we recognize places based on vision, would grid cells also activate, or otherwise would that mean that?
Yeah, I mean, there's a lot of work that is nowadays being done in virtual environments where especially mice run on wheels or on balls, and then you can dissociate the types of input. For example, the locomotor ones, which are the ones that are much easier to study in our studies. But you can also provide input through optic flow, the running of the visual image as the mouse gets forward in time, and you can do various tricks to dissociate the optic flow from the actual locomotion on the ball. And then this is mostly work done in London, so again, correct me, Neil, but what I take from that is that they both matter. You both generate place fields from the locomotion, but also from the passage of particular cues, and that is sort of, there must be some redundancy, but maybe some cells rely more on one and some cells rely on other, but it's not exclusive. Is that correct?
I think that's right. Most of the cells require both inputs, but there's about 25% that could fire just with one or just with the other in terms of plate cells.
Yeah.
Oh, a very quick question. Your spatial non-grid cells at the end there, did they include border cells?
They do not. I think they are not border cells in the traditional sense, because then they would, in that environment, have a strong field along one of the wall. It's a bit more hard to say whether they correspond to what you call boundary vector cells, because then it's harder to measure, in the sense that they then fire out in the field, and the field would be more blurred than if it would be in the environment. But related to that question, one absolute possibility is that these cells may also be cells that fire in relation to objects and have object vector fields when an object is present. But then in an open environment, I mean, there isn't any discrete punctuate object, but there are still walls and maybe distal inputs, which perhaps may create more variation in the actual firing location. So I think we can't totally yet dissociate them from those. It's possible that object vector cells and also maybe even border or boundary vector cells might be in that second group. But it's not the traditional border cells, because we also investigated border cells and head direction cells with these two manipulations. And turning off parvalbumin or somatostatin interneurons has absolutely no effect on border cells or head direction cells.
Okay, I have actually two questions. You mentioned that grid cells were completely maintained under sleep. Do you have data under anesthesia? And
So what did I say? I didn't get that.
does anesthesia preserve the grid cells?
Oh, yeah, not anesthesia, but sleep anyway, yes.
So there's no data during general anesthesia?
No, I haven't. I think it probably depends on the level of anesthesia. At some point, I think everything would break down. But I think the pattern, I would still think that at some level of anesthesia, or light anesthesia, or maybe urethane anesthesia, or whatever, that you might preserve it, because I think so far we haven't been able to see any intervention that interferes with the actual correlations or cross correlations between cells.
And
but it depends. I mean, if you turn off the whole system, of course, it wouldn't work anymore.
Thank you. My second question was, do you have some microstimulation examples or accusations? Can you, for example, induce border effects artificially?
Yeah, no, that type of experiment is what we want to do. So I think the day when we find a molecular marker that is specific for grid cells or specific for border cells, of course, then we can turn them on and off as we want. We are not there yet, but I'm sure within the next 10 years, someone will be able to do that. Okay, we should monitor the time, but I guess you do that, Paul, because I think I'm much over time, really.
No, we're okay, don't worry. Diogo.
So regarding speed representation in the internal cortex, so if you look at hippocampal CA1, you can actually find that representation being provided by the LFP, specifically theta modulation correlates to the speed or the running speed of the animal. From a modeling perspective, why do you think is needed to convert that LFP representation into a firing rate representation for the medial internal cortex?
Well, I'm not really sure I would consider it a conversion. I mean, you have. I think it's, to me, it sounds like it may be probably a reflection of the same, that you have the LFP. That is the relation between LFP and speed is reflected in because there is rate changes that depend on firing rate. So I think the way we have tried to handle it so far is to try to find out where does this speed modulation comes from, and it seems to depend on inputs that are from the medial septum, which again depend on inputs from the mesencephalic locomotor circuits. So and probably is expressed widely or over the brain. But then, in order to understand how this rate change depending on speed is actually generated, I mean, you see the same even in the mesencephalon speed cells. So I think it is probably generated at a very low level of the brain, and then just maybe just broadcasted over large parts of, including cortex.
Lisette.
Yes, so I want to ask about your view of how this grid pattern could appear in a space, but also in time.
Yeah.
Because what you have shown during a sleep is
Yeah.
basically a temporal grid pattern.
Yeah.
Now, if this happens when the animal is not moving, this means that you have an internal dynamics that determine the temporal relationship. Now, because time and space are linked when you move, my question is whether you think that the path integration framework can explain, for instance, experiments as you see when you have an empty environment and you have the grid, but then you have these corridors, and the grid break up in stripes that appear, and the animal is moving the direction of the movement. So path integration will accumulate errors and will not sustain that. Only an internal dynamics could explain that. So I would like you to comment on that and see what is your point of view on this.
Well, there is representation of time too. That's just not at all so well understood. But and as you know, there are at least two indications of representation of time. So first is that if you record over long times, especially for some reason expressed very well in CA2, there are actually changes that go on, so that if you decode from the population activity the actual time, you can, and you will see that it changes much more than over time than in other regions. And now we show that the same actually is the case in the lateral entorhinal cortex as well. And of course, then in addition, you also have time cells, which I mean, there's been a lot of discussion whether they actually just reflect path integration or not. But I mean, Eichenbaum did the experiments which showed that you can try to dissociate them and still have a time component expressed in these cells. I think we still don't understand that very well, but I think there is a time component that is superimposed on the spatial.
All right, fantastic. So thank you very much, Edvard.